Can you start? Good morning, students. Whether I'm audible? Good morning, everyone. Good morning. Just a reply whether I am audible or not. Good morning, Sweta. Yes. Yes. Good morning, everyone. I hope your preparation is going well. Okay, just 1 month is there. Give your 100% effort. Okay, today we are going to see previous year question papers. Okay, we will start with 2025 up to 2020. We are going to discuss. Okay, today we are going to see about 2025 previous year question papers. Okay, just be interactive in the class. Then comment the answer in the comment section. If you have any doubt, you can ask there itself. Okay. First question, given below are the two statement, one is assertion and another is labeled as reason. Okay, just read the question, what they are giving. Rubisco enzyme is the most abundant biological catalyst on the earth. It accounts for half of the soluble protein content in the chloroplast. Whether assertion is correct? Then reason they are saying, one of the reason for Rubisco's high concentration is plant chloroplast. Okay, plant chloroplast have this one. Then it is partially efficient enzyme. What will be the correct answer, students? What will be the correct answer? Yes, C. Okay, someone is saying C. Anyone else? Udaya Lakshmi saying C. Yeah. Read the question properly. Assertion is right only. Rubisco. Yeah, Rubisco is, yeah. Kencom, correct answer. Option A is the right answer. Why we are going to discuss now. See here, Rubisco is the most abundant biological catalyst. Catalyst means what? Nothing but an enzyme. So it's the most abundant enzyme on the earth. Okay. Even though it is most abundant, the catalytic efficiency of this enzyme is less. Why means each molecule of enzyme will fix only 3.3 mole of CO2 per second. Okay, so because of this less efficiency, this enzyme abundance will be more in the chloroplast to convert the carbon dioxide to sugars. Okay, so the right option is both A and R are true, and R is the correct explanation of A. Okay, then identify the crops where tryptamine pathway of auxin biosynthesis is found. What will be the answer? During our class itself, we have seen there are 4 different pathways are there for auxin biosynthesis. 4 different pathways are there. Yeah, tomato, okay, excellent. Any other else? Option C. Excellent, students. Tomato is the right answer. T. See, there are 4 different pathways of auxin biosynthesis are there. Okay, what are the 4 pathways? That is tryptophan pathway, indole pyruvic acid pathway, indole acetamine pathway, and indole acetonitrile pathway. There are 4 different pathways are there. Okay, this tryptamine pathway is most common in tomato, like these crops will have this tryptamine pathway. Okay. Indole pyruvic acid will be there in peas. Okay, peas like this, common pathway. This is clear. Indole acetamine pathway will be an bacterial pathway. It's an bacterial pathway because we know this Agrobacterium tumefaciens also will have this oxygen biosynthetic genes. Okay. So this bacterial pathway is known as indole acetoamine pathway. Okay. Then indole acetonitrile pathway will be there in 3 species. Okay. What are the species? That will be there in mustard. Okay. In mustard, that means crucifer families it will have, Brassicaceae family it will have. Then rice will have this in IAN pathway. Apart from that, banana also will have this IAN pathway. You just remember this one. Okay. What are the different pathways and What are the major example for each and every pathway? Okay. Next question. A protein is likely to bind tightly to cation exchange resin. They are asking about ion exchange chromatography. Cation exchange resin under which of the following condition? This we have discussed in our class itself. What will be the answer? Protein is likely bind to cation exchange resin. Okay. They have given different things. What will be the buffer concentration? How the pH and pA will differ? You are getting the question. Any answer, students? Yeah. Any answer? We know the basic concept. If pH, yeah, someone is saying B. Yes, B is the correct answer. B is the correct answer. Excellent. We will see how. Okay. We know the basic concept. If pH is greater than pI, what will be the charge on the protein molecule? If pH is less than pI, what will be the charge on the protein molecule? If pH is less than pI, that molecule will have positive charge. This we already know. If pH is greater than pI, that molecule will have negative charge. Then this ion exchange chromatography, there will be two different resins will be there. Okay. What is that? Cation resin or anionic resin? Okay. Or anionic resin will be there. Okay. These are the resins. So cation exchange chromatography, in cation exchange chromatography, it will binds the cations. Okay. So the resin will be negatively charged. Okay. Then anionic resin means that will bind the anionic molecules. The resin will be positive charge. Here they are asking a protein is likely to bind to cation exchange resin. Okay. This is cation exchange resin. Under which of the following condition? So it is clear the protein should have positive charge. So pH should be lower than its pI. So pH is lower than its isoelectric point of the protein. I hope this is clear. Then second thing, what? Buffer concentration should be less so that it can easily go and bind with this resin. I hope this is clear. Third, they have given different vitamins along with their coenzyme form. Just match it one. What are the different vitamins and what are its coenzyme form? Yes, vitamin C, what will be the example for that? Coenzymic form? Vitamin K, vitamin B12 and lipoic acid. Yes, what will be the answer? Vitamin C, it's most important for what? Hydroxylation of specific proline residues. This is the most important function. Yeah, option A. Okay. This will be the most important function of vitamin C. Vitamin K, what it will do? Blood coagulation by gamma carboxylation of specific glutamate residue. This also we have discussed in our class. Okay. Vitamin B12, the specific function of vitamin B12 is what? Methylation of homocysteine it will do. So this will be the C. Lipoic acid, this we already seen in pyruvate dehydrogenase complex. This will act as one of the cofactors. So oxidative decarboxylation of alpha keto acid. So this will be the, what will be the right answer? A will be 3, B, C and C for 2, D for D. So the correct option will be 3, 4, 2 and 1. I hope this is clear. Okay. What are the epigenetic modification? They have given different changes. We have to pick out which one is the epigenetic modification in plant. What will be the epigenetic modification among this one? Addition of, yeah, correct. They are saying the addition of reversible changes to histone protein and DNA is one of the epigenetic changes. Okay, yeah. See, so epigenetics means what? We already know epigenetics. Epigenetics, that means what? The sequence of the genome or the sequence of the gene will not be changed, but there will be an reversible changes. Reversible changes will be done in the bases or with the histones that will influence the expression of this particular gene. Clear? Influence the expression of particular gene. Okay. without changing the nucleotide sequence. Okay, how the reversible changes in the histone as well as in DNA molecule like methylation, like these things influence the gene expression is known as epigenetics. Okay, this is clear. Next, carbohydrates can be purified by affinity chromatography. Okay, using which of the following immobilized protein? They are asking about affinity chromatography. They have given here different ligands they have given. Okay, different ligands they have given. How these ligands, which of the following ligand can be used to purify the carbohydrates? Any guesses? Techniques in biochemistry. Yes, in this section we have seen this one, technique in biochemistry. Yes, both concavalin A and wheat germ gluten. This is correct. Yes, concavalin A and wheat germ gluten or agglutinin are commonly used for what? Carbohydrate separation or purification. Histidine tags. Okay, histidine tags will be used for nickel purification. This is another important question. Okay. Then protein A will be used for immunoglobulin separation. Okay. Or else we can use particular enzyme to isolate the particular substrate or cofactor, or else we can use particular antigen to isolate the particular antibody. These are the different ligands that are used to purify the particular components in a mixture. Okay. Heterosis increases the level of what? General Agri question. Heterosis increase the level of, whether it will increase homozygosity or heterozygosity or polyzygosity, which will be increased in heterosis. Yes, heterozygosity will be increased in heterosis. Okay, heterosis is nothing but it's an hybrid vigor. The performance of the plant will be more comparing to its parental. Okay, so it will increase the heterozygosity in heterosis. Okay, if this is another important question. Okay, if all the nucleotides are present in equal frequency, just read the question. If all the nucleotides are present in equal frequencies, that means 1 out of 4. This is the equal frequency. Adenine, guanine, thymine and cytosine. They are saying these are present in equal frequencies at and random. What are the chances of having a particular form of nucleotide long motif? That is 4 nucleotide long motif is there means what will be the probability? Okay. Yes, excellent. 1 by 256. See, simple. They are asking about 4 nucleotide. Okay. If 4 nucleotide stretch, for example, that may be ATGC or CGAC. Like this, different formation will be there. They are asking what is the probability. Simple. 1 by 4 is the 1 by 4 whole power n. So, 1 by 4 whole power of it is an tetranucleotide. So, 1 by 256. I hope this is clear. Like these questions, they can ask. Problem solving question, they can ask. Okay, so 1 by 256 is the right answer. Okay, you all know about our academy. Okay, if you want to take our courses, okay, now price has been dropped to 1999 rupees. We have covered all the aspect of plant biotechnology. Just join our classes. Then mock test series is also there with us. Okay, then kindly join for the mock test series also. Then for ICAR 2028 and 27, our classes are going to come. Okay, kindly join that also. Okay. Next question. Here they have given the different restriction enzyme. Okay, they have given different restriction enzyme. Then they are asking what will be the sites. Okay, after cleaving, what it will form, whether it will form a protruding end or blunt end. Like this, they are asking. This we have seen in our lecture itself. Can anyone guess what will be the answer? PST1, what it will be produce? Hay 3, Eco R5 and Hind 3. What will be the right option, students? Yes, what will be the right option? Hey3, Eco R3, PST and Hind 3. Any guesses? Any guesses? Any guess? Yes, I will wait. Any guess? B. Option B. Okay. Any other guesses? Any other guesses? Any other guesses? Hmm. Option B. Yes. Option B is the right answer. Okay. See, PST3 will produce 3' protruding end. This we have already seen. Okay, PST3 will produce 3' protruding end. This we have already seen in our class. This is one of the most important restriction enzyme. Okay. Then He3 is a 4-base cutter. This we already discussed in our class. Simple question. He3 is a 4-base cutter. Eco R5 is an blend 10 producing restriction enzyme. But Eco R1, Vinutaraj 2025 paper solution is given in test series, answer is not matching. Kindly like post this question here. Okay. See, Eco R5 is it's producing blend end. Okay, blend end it will produce. Then Hind 3. This Hind 3 will produce 5' protruding end. Okay, this is clear. This is clear. Okay. Next question. See here I have given all the restriction enzyme along with the organism from where they have isolated. Then what is the recognition sequence and whether it will produce blend end or sticky end we have given here. Okay. Then here they have given the different complexes of electron transport chain. Okay. Match the following. What is the different complex? Complex 1, 3, complex 5 and complex 4. And they have given the specific of that each complex. Okay, we know this complex 1 is the what? It's the largest complex. This we already know. Okay, complex 1 will be the largest complex. See, largest electron carrier protein in the mitochondrial respiratory chain. This is right. Yeah, first one will come option A. Okay, then second one, complex 3. What it will do? This complex 3 will have what? Two cytochromes will be there. This is clear. Complex 3 will have cytochrome B and C1, cytochrome B and C1 will be there. Then risky ion center will be there. So for complex 3, B will be the answer. Okay. Then complex 5, that is what? ATP synthase complex. It will have F0 and F1 component. This we have already clearly seen. Membrane will be there. This F0 will be embedded in the membrane and F1 will be there in what? Matrix side. So what they are saying? So F1 component is what? It is an hydrophilic component. Okay, it's an hydrophilic component. The right answer for this, yeah, affinity chromatography, immobilization, the both the things will come. Okay, just you change that one. Right answer is concavalin also will come. Wheat gem agglutinin also will come for purifying the carbohydrates. Okay. Vinutaraj, just you change that options. Okay. That is the correct answer. Whatever we are discussing in our live series, this is the correct answers. Okay. If any mistakes are there, just rectify that one. Okay. See here. So complex 3 will, complex 5 will have hydrophilic domains. That is F1 domain. Then complex 4, what it will do? It will translocate the H plus ions from matrix to intermembrane space. Two molecules of H plus will be transferred. Okay, so the right option will be option A. Clear? There are 4 different complex in electron transport chain. Complex 1 will pump 4H plus. Complex 3 will pump 4H plus ions and complex 4 will pump 2H plus ion to intermembrane space. This we have discussed already. Okay, clear? Next, after the catalyzing the conversion of succinate into fumarate, the FAD prosthetic group of succinate dehydrogenase enzyme get reduced to FADH2. Okay, the reoxidation of this enzyme FADH2 is carried by which of the following molecule? Any guesses? Which of the following molecule? Any guess? They are interrelating TCA cycle with oxidative phosphorylation. TCA cycle with oxidative phosphorylation they have given here. Uday is saying TPP. Any guesses? Observe clearly, succinate dehydrogenase enzyme, TK, it will be, yeah, ubiquinone. Ubiquinone is the correct answer. See, succinate dehydrogenase, what it will do in electron transport in citric acid cycle, here FADH2 will be produced. TK, in mitochondrial electron transport chain, this FADH will donate the electron to where? Here complex 2 will be there. TK. From here, the electron will be accepted by ubiquinone. So, for reoxidation of this FADH2, ubiquinone will act as an ultimate electron acceptor. So, this is the right answer, ubiquinone. TK. TPP will come as a cofactor in alpha keto acid decarboxylation. This we already seen. TK. This is clear. So, FADH2 reoxidation will be done by ubiquinone and complex 2. Okay. Then they have given here different tools, bioinformatic tools along with their functions. Okay. See, simple thing. You have to match among these all the things we know. Sequence homology tool. This is very basic tool. BLAST we know. For sequence homology, we will use BLAST. So first one will come option 3. See here, only in this option A is option 3. So this will be the right answer. Next thing also we can see motif. Motif tool will be helped for some of the motif tool is what? Copia. Copia is an example of motif tool. Yes. Information retrieval tool. Entries is the one of the major information retrieval tool. Multiple alignment tool. This is another most important question. TK. They have asked one time in like choose the best answer. TK. Multiple alignment tool example is cluster W. Cluster W is example of multiple alignment tool. TK. See the next question they have given different plant hormones along with their major function. Different plant hormones along with their major function. Gibberellins, what it will do? Majorly it will increase the alpha amylase production during seed germination. So first will come option 4. Anything is there? Yeah, this will come. Yes. Then second is what? Auxin. The major function of auxin is what? Induction of, any guesses? Induction. No, cell elongation will do. Cytokinin. Cell division. Cytokinin is the cell division hormone and ABA is what? Stomatal closure. They have given clearly here. We can select the option by elimination method. Okay, so A is the right option. Okay, any guesses? They are asking about Mendelian P experiment. They have given two statements. We have to answer which one is right and wrong. Okay, statement 1 is Mendelian P experiment. See here, wrinkled seeds. Seed is wrinkled due to mutation in a gene encoding starch branching enzyme. Okay, they are saying the starch branching enzyme is mutated because of this wrinkled seeds are forming in Mendelian experiment. Okay, then they are giving another statement. Wrinkled seed of pea accumulates a large amount of starch. See here, what will be the right option? They are saying in the statement 1 mutation is there in starch branching enzyme. Then how high amount of starch will accumulate? So statement 2 is ultimately wrong. Okay, if the gene is, if the starch branching enzyme is mutated, how there will be a high accumulation? Yeah. So A is true and B is false. Excellent. A is true and statement 2 is false. Okay. Another simple question under direct question. The process by which a viable seed resumes growth under favorable condition. Under a favorable condition, the seed will resume its growth. What will be the option, students? What will be the answer? Under favorable condition, the seeds will germinate like that. They are saying. What will be the answer? Yeah. Germination. Yes. Germination means the seed will resume its metabolic process to growth. Yes. Germination. Okay. Then stratification means what? Stratification means We are giving cold treatments for promoting seed germination. Okay. Scarification means what? Scarification. Okay. It's a mechanical method. Okay. Yeah. It's a mechanical method. Scarification means it's a mechanical method. Just we will do this for loosening the seed coat of seed. Okay. So that it will facilitate the easy uptake of water and germination will be proceed soon. Okay. Next, this is another important question. They have given an sequence of proteins, okay, different amino acid sequence they have given. Then they are asking here, what will be the charge of this peptide? They are asking what will be the charge of the peptide. Then most important thing what they have given in this question is they have given pKa, okay, pKa for each and every amino acid. You see here they have given pKa for N terminal, that is pKa for alanine they have given, pKa for C terminus, glutamine they have given, pKa for arginine and pKa of aspartate they have given here. Okay, you think properly, but they are asking the question, what will be the charge at the peptide at physiological pH, pH 7.4. In this pH, what will be the charge of this peptide, like that they are asking. So, we have to consider the physiological pH as well as pKa of the amino acids, whatever they have given. Based on that, we have to calculate here. So, we are going to solve this. Just listen properly. Alanine, arginine, valine, aspartate, and glutamine is there. Simple thing first. Among this amino acid, which will be having the charge? Aspartic acid will have negative charge. Glutamic acid also will have negative charge. Arginine will have what? Positive charge will be there. Positive 1 plus 1 will be there. Okay. Okay, this is clear. So this is clear. But you see here, whatever the pKa given for N terminus, C terminus and arginine and aspartate, if you see, start from N terminal. The N terminal pKa is 9.0, but pH is 7.4. Okay, pH is 7.4, but pKa is what? 9.0. So there will be an influence of positive charge on this alanine, okay, because of this pH difference. Okay, there will be a one positive charge will come. Here there is no influence will be there because this valence structure we know it's an amino acid where the carboxy group and amino group will be involved in the peptide bond formation. So here charge will be 0. You just add this plus 2 minus 2. So 0. The net charge on the this polypeptide, given polypeptide will be what? It will be 0. So the simple method is first you have to calculate what are the positive charge amino acid and negative charge amino acid present in the given peptide, and if they given the pKa value for each and every amino acid that we have to consider, then we have to calculate the pKa of that amino acid. I hope this is clear. Okay. This is another important question. They have given different restriction enzymes. Okay, sorry, different enzymes that are used in genome editing technique. This we have already discussed in our class. What will be the right answer? They have given Cas9, Fock enzyme, ICAR, then different restriction enzymes. Then what are the activities or features? Yes, Kankam is saying option D. Any other answers, students? Any other guesses? Yes. We know Cas9 that will be primarily used, yeah, primarily used for CRISPR-Cas genome editing technology. So Cas9 will be primarily used for CRISPR-Cas genome editing technique. Fork1 endonuclease will be widely used in zinc finger nuclease and talons. This we have already seen. Zinc finger nuclease or talons. Eco R1, it's a restriction enzyme that will produce sticky end at 5' end. Okay, 5' end sticky end will be produced. ALUI, blunt end production will be there. So the right option is what? A2, B3, C1 and D4. So option D is the right option. Yes. Next. Who proved that DNA was indeed the genetic material through experiment? They are saying DNA is the genetic material was proven experimentally by whom? DNA is the genetic material. Yes. Who proved? Genetic experiment. Yes, any guesses? Any guesses? Genetic matter. Yes, D. Excellent. Hershey-Chase experiment. In Hershey-Chase experiment, they have proved that DNA is the genetic material of an organism. Then, who proved? Who has proven RNA is the genetic material? This is homework for you guys. Prove RNA as genetic material in viruses. You just go through this one also. Who has proven, or experimental proof who has given for RNA as a genetic material. Okay, this I will ask in the next class. Okay, this is homework for you. Okay, just you see who has proven RNA as a genetic material in viruses. Okay, next question. Which of the following is an example of, see here, horizontal gene transfer? They are asking horizontal gene transfer example. Okay, read the option properly. Transfer of antibiotic resistant gene from one bacterial species to another bacterial species. So this is one bacterial community or species. Here is there. So antibiotic resistant gene is transforming from one species to other species. Second option is transfer of gene from one bacterium to another member of the same bacterial species. Okay. Option C, transfer of antibiotic resistant gene from one bacterial to fungal species. This will never come. Okay, that will comes under vertical transfer, like that also we can say, but this is theoretical not possible. Okay. Then transfer of gene from bacteria to fungus. This also will not come. So the right option is what? Transfer of antibiotic gene, resistant gene from one bacterial species to another bacterial species. This is known as what? Transformation we will do. Correct na? In our gene cloning and recombinant DNA lecture, we have seen these things. Okay. We have seen the plasmid that will be transformed from one bacterial species to another bacterial species. Okay. So that is an example of horizontal gene transfer. Okay. So the right option is option A. Leaf area index is defined as, plant physiology question. In our last class, we have already discussed the different parameters, growth parameters used and what is the importance, units, everything I have discussed in the class. Okay, leaf area index is defined for what? What it will show? Yes, any guesses? Leaf area index. Whether it will show total leaf area per plant or leaf area per unit ground area. This is most repeated question. Yes. Whether it will show leaf dry weight, leaf area per plant, or leaf area per ground number. Yes, excellent. It will show leaf area per ground area. Leaf area per ground area. Okay. You see this properly. This is most important table. Okay. See here. Leaf area index is what? Ratio of leaf area to ground area. Then that is unitless. Okay. This is another important thing. That is unitless. Okay. You just go through this all the formulas along with the purpose why we are using this growth parameters. What is net assimilation rate, crop growth rate, RGR, TK, leaf area ratio, specific leaf weight. What is the purpose? You just go through this one. From this, they will sure ask one question. TK. So this is very important topic. TK. Next, they are asking about libraries, genomic library, cDNA library, like that. They are asking question about library. See here, which of the following type of library has chance to carry the maximum representation of protein coding genes of total number of genes? Tell students, they are asking protein coding genes will be there in which library? Yes, protein coding genes. Genomic library or cDNA library or SIR? Yes, cDNA library will have what? Protein coding sequence will be there. How? We will isolate the mRNA, messenger RNA. This we will convert into complementary DNA, that is cDNA. This will be cloned in vectors. So, these cDNA clones will have the protein coding sequence. Majority of the protein coding sequence will be There, yes, clear. The proportion of DNA that encode ribosomal RNA are collectively referred to as what? Proportion of DNA that encodes RNA are collectively referred to as, whether it is a nucleolus or nucleolus organizer region, it's a centromere or centriole. What will be the right option? Yes, nucleolus organizer region. Nucleolus is an what? It's an organelle where ribosomal RNA synthesis will happen. But nucleolus organizer region is what? It's a specific region in the DNA that will codes for ribosomal RNAs. Okay. Next, they have given about fatty acids. Okay. They have given about arachidonic acid and they have asked multiple statements. Okay. You know, arachidonic acid is a what? It's a 20 carbon fatty acid with 4 double bonds will be there. Okay. That will be present at 5th position, 8th position, 11th and 14th position. Okay. This is clear for everyone. I know this will be very easy. It's a polyunsaturated fatty acid that contains 20 carbon atoms and 4 double bonds. Yes, this is correct option. Then you see here, they have given both omega 3 and omega 6 for same fatty acid. So among them, any one of the option will be wrong. Okay. So you see here the position of double bond, it is there at 5, 8, 11 and 14. But the fatty acid length is 20. So 20 minus 14, last double bond is 6. So this is an omega 6 fatty acid. So the right option is it's an omega 6 fatty acid. It's not an omega 3 fatty acid. So wherever B option is coming, that all are wrong. So see here, here B option coming, it's wrong. Here B option coming, that is also will be wrong. So A and C is right. Okay, so A and C is coming here. This will be the right option because D they have given in all the option. Clear? This is elimination method. You have to work smartly also. Wherever our right option is coming, that you check first. Wherever wrong option is there, that we have to eliminate. Then we have to pick the answer rightly. Okay. We know this arachidonic acid is an essential fatty acid. Yes, it's correct. From this essential fatty acid only, different signaling molecules will be produced. This we have already discussed. Prostaglandin, thromboxanes, leukotrienes, these all the compounds will be synthesized from this arachidonic acid. This we have already discussed. Okay. Next, different alleles of a gene, different alleles of a gene produce difference in observable, see here, produce difference in observable features of an organism. The future is known as, what will be the future? They are saying here, it's an observable feature. Okay, that will be a visual feature. Then whether it will be a genotype or phenotype, excellent, phenotype. It's a phenotype, okay, because observable feature means that will come an phenotype. Okay. Next, they have given the different terms. Okay, then what will be the function? Photoperiodism, transpiration, respiration, stomata. Very simple question they have asked. See, photoperiodism, this we already know. It is what? It will, how the light will affects the flowering, that we can study. The effect of light duration on flowering. So option A will come 4. Option A will come 4. Yes, option A will come 4. Transpiration means what? There will be an what? Loss of water molecules will be there. Loss of water from the leaves. Respiration. This we know. The complex carbohydrates and sugars will be break down to produce energy. So breakdown of glucose to release energy. Stomata. The major function we do that will be involved in opening and closing of the guard cells. By that action, what carbon dioxide can come inside and water molecules can go out of the plant. So option B is the right one. Okay. Next question. They are asking about C3 and C4 plants and how it is influenced by light. Okay. C3 plants grown more slowly. They are saying C3 plants grow more slowly than C4 plants when light is limited. If light is limited, they are saying C3 plants will grow more slowly. Okay, whether this is true? No. C4 plants only will requires more light. Why? Because the energy consumption for each molecule of CO2 fixation will be higher in C4 plant comparing to C3. For example, 5 molecules of ATP will be required for fixing per molecule of CO2. But here only 3 molecule of ATP will be required. So the first option will be, first statement is wrong. C3 plants will grow more fast than C4 plants. That is the right option. Okay. Second, C4 plants requires more sunlight to meet the high energy demands of the C4 pathway. Yes, this is correct. So the right option is statement 1 is incorrect and statement 2 is correct. Clear, students? Yes. Yes, can come clear. Many. Next, observe the question properly. In floriculture, gibberellins are often used to. For what purpose they will use? Gibberellins are often used to. Floriculture, they are asking. Seed germination or induce early flowering or promote root growth or inhibit flowering. Yes, it will. They will use for what? Induce early flowering. Okay, induce early flowering. They will do bolting. Bolting is the major function of this gibberellic acid. Okay, yes, clear. Yeah. The hormone that used to kill dicot weeds. What will be the hormone? Plant hormone that will be used to kill dicot weeds. NAA or NBA? Yes, 2,4-A. 2,4-diclonophenolic acetic acid will be used as an what? At higher concentration of this auxin that can be used as an weedicide to kill the dicot weeds. Apart from that, another important thing is there, thiodiazuron. Okay, thiodiazuron. Okay, it's a synthetic cytokinin. Okay, it's a synthetic cytokinin. This can be also used for defoliation of the plant. Okay, clear? So thiodiazuron can also be used as an defoliant. Okay, clear? Next, TY element. TY. What is this TY? First, we can see what is the TY. TY means what? It's a transposable element. Okay, transposable element in where? In yeast. They are asking this transposable element is an example of whether it's an LTR containing transpose. LTR means what? Long terminal repeats. Okay, long terminal repeats. This TY, whether it will have long terminal repeat or this will not have long terminal repeat, or it's a class of TN3 type transposon, or it is a DNA transposon, like that they are asking. Okay. Okay. What will be the right answer? Yes, it's an LTR containing retrotransposon. Why it's an LTR containing retrotransposon means this has to be successfully integrated to our target region of the genome. TK. TK. This will has to be successfully integrated in the target region of the genome. For that, this long terminal repeats plays a significant role. That's why this TY element in yeast, it's a specific example of LTR containing retrotransposon. Okay, LTR containing retrotransposon. See here, another question, they have given different enzymes that are involved in different metabolic pathway, along with that major role in fatty acid metabolism. Anyone can answer? ATP citrate lyase. ATP citrate lyase, what it will do? As we know, the beta oxidation will happen in mitochondrial matrix. The acetyl-CoA produced after beta oxidation or whatever the oxidation process happen, that has to be shuttled to cytoplasm for fatty acid synthesis. There comes the role of what? Citrate cycle will happen there. Okay. So there what it will come? ATP citrate lyase will plays a primary role for shuttling the acetyl-CoA from matrix to where? Cytosol. So facilitate the transporter of acetyl-CoA. That means that is the precursor from mitochondrial matrix to cytoplasm. So this will be the option A. Okay, wherever is coming this, so here only. Yes, yes, Udaya, correct. So HMG CoA lyase, they are saying HMG CoA lyase. This we have already seen in our ketone body synthesis and degradation class. Okay, HMG CoA lyase will be primarily involved in synthesis and easy transportable ketone bodies. Okay. Thioesterase and enoyl-CoA isomerase. This also we have seen. Thioesterase, what it will do? The fatty acid, once it's synthesized, that has to be removed from that enzyme, that has to be released from that enzyme complex. There comes the role of thioesterase. So, what it will do? Termination of fatty acid synthesis will be, facilitate the degradation of unsaturated fatty acid will be done. Then enyl-CoA isomerase, we already seen this one. Enyl-CoA isomerase, what it will do? There are two different enzymes are there that are specific for degradation of unsaturated fatty acid. What are the two enzymes? Del 3, del 2 enyl-CoA isomerase, and 2,4-dienyl-CoA reductase, dienyl-CoA reductase. Okay, this we already discussed in our lecture series. Okay, so the right option is option B. Clear? Next, for increasing the vast life of cut flower, the holding solution pH should be in the range of, they are asking if we have to increase the shelf life of the flowers, the holding solution should have an pH of what? Whether it should be an neutral pH or alkaline pH or more acidic or optimum pH, like that they are asking. Okay, this is direct question. Okay, general agree question. See, the pH should be around 3 to 5. Okay, pH should be around 3 to 5 for the solution. Why it has to be around 3 to 5 means, first thing, bacterial growth will be inhibited at low pH. Okay, bacterial growth will be inhibited. Then second thing, comparing to alkaline solution, okay, acidic solution uptake by these cutted stems will be more, so that flowers will remain fresh. Okay, that's why they are using pH 3 to 5 for this solution. Okay, clear? Next, if the water potential, another important thing, if the water potential of a solution outside of the cell is -1 megapascal, they have given. This is cell outside. Then water potential of the cell sap inside the cell is -1.4 megapascal. Okay, inside cell. Then from where to where water will move? From cell to soil it will move, or from soil to other outside it will move. What will be the answer? What will be the answer? Yes, we have already discussed these things. Water will always move towards its more negative water potential. Yes, diffuse into the cell. Correct answer. Yes, diffuse into the cell. See here, comparing to -1, -1.4 is the more negative. So water will move from outside to inside the cell. That is diffusing into the cell. Yes, clear. Next, C4 plant, commonly cultivated C4 plant. Simple question, direct question. Example: wheat, potato, sugarcane, rice. What will be the example? Yes, sugarcane will be the right option. Next, identify the aerobic nitrogen fixing bacteria. They are asking about aerobic nitrogen fixing bacteria. Okay, aerobic nitrogen fixing bacteria. As we know, Clostridium. Clostridium is an example of anaerobic. This we already seen in our lecture. Clostridium is anaerobic, so C will never come. Rhodospirillum is also an anaerobic. Yes, Rhodospirillum is also an anaerobic. So right option is A and D. Yes, clear student. Astobacter and Gliotheciae is an example of aerobic nitrogen fixing bacteria. Anaerobic nitrogen fixing bacteria is Clostridium, Rhodospirillum and Methanococcus. Okay. Clostridium, Rhodospirillum and Methanococcus are example of anaerobic nitrogen fixing bacteria. Okay. They have asked about CRISPR-Cas genome editing. The CRISPR-Cas associated Cas9 is one of the important component of CRISPR, adaptive immune system in Streptococcus pyogenes. It is an what? It's an RNA guided endonuclease or DNA guided DNA exonuclease or RNA guided exoribonuclease or DNA guided exoribonuclease. What will be the answer? Yes, RNA guided DNA endonuclease. Yes, excellent. See, there will be a guide RNA. Here, Cas complex will be there. This will go and make double stranded break at target DNA. So, it's an RNA guided DNA endonuclease. Yes, very good, students. Next, in the process of transcription, RNA is complementary to. Read the question properly. In the process of transcription, RNA is complementary to what they are asking. RNA will be complementary to what? What it will be? It will be complementary to template strand or coding strand or DNA strand or coding strand having 3' to 5' orientation. Yes, see DNA stranding having 3', 5' to 3' orientation. Okay, any other guesses? See, this is the double strand of DNA. 5' to 3' will be there. Then 3' to 5' will be there. This 3' to 5' will always act as a template for transcription. So, by using this template, the complementary nucleotides will be added here. Okay, so 5' to 3' will be there. Correct. 5' to 3' will be there. So, this will be similar to 5' to 3' of the what? Similar to 5' to 3'. So, this is wrong answer. They are asking complementary. So, it will be complementary to what? This will be always complementary to, yes, Kenkam. This is clear. They are asking which one will be the complementary thing. This will be complementary to what? It will be complementary to template stand of DNA only. It will be complementary. Okay. C, D, coding stand. Coding stand is same only. Coding, see, you first remember these things. Coding stand means what? That will code for the protein. Okay. So the information will be present in the 5' to 3' code. Okay. Yes. So this is the coding stand. This is the template stand. Template stand means what? It will be involved in the synthesis of this transcription. That means messenger RNA will be synthesis. They are asking complementary. RNA is complementary to what? They are not asking similarity. They are asking complementary. It will be complementary to the template stand. TK, complementary to the template stand. This is little much tricky question. You have to answer properly. TK. Identify the synthetic cytokinin commercially used as an defoliant and herbicide. This we have already seen. Yes, thiodazole will act as an defoliant and herbicide. Okay, it's a synthetic cytokinin. Excellent, students. See here, they have given about nucleides. What is the characteristics of nucleides? And they have given about some of the functions of these E. coli genetic materials also, how it will be there like that. Okay. This is a simple question. I know you will answer, guys. Nucleide is the characteristics of eubacteria. This is true only. Escherichia coli contains its genetic material present as a circular DNA molecule that is compacted into an unenclosed region. What will be the answer? What will be the answer? Yeah, both 1 and 2 are true. Excellent. What happens when double-stranded DNA virus enter into the plant cell nucleus? What happens when a double-stranded DNA virus enters into a plant nucleus? What it will happen? Whether it will remains as a double strand or whether it will be converted into mRNA or single stranded RNA, what will be the answer? From, see here, if for example this is a cell, if an virus is infecting this one, the double stranded RNA will be there. Okay, it will double stranded DNA will be there. This will always remains as double stranded only. How? It will just go and integrate in the genomic DNA region. Then this will use host machinery for doing all the processes. Okay, replication, everything will be done by using this host machinery. So the right option is what? It remains as a double stranded DNA only. It will never form a single stranded DNA, or never it will form single stranded RNA or single stranded DNA and RNA helix. Okay. So the right option is it will remains as double stranded DNA. Okay. Next, what are somatic hybrids? From tissue culture they are asking. What are somatic hybrids? Any guesses? Somatic hybrids. Hybrids. It's a protoplast, or a hybrid protoplast, or fused plasmid, or a fused chloroplast. Yes, excellent. It's an hybrid protoplast. See, protoplast from two plants we will isolate, then we will fuse these two from this somatic hybrids. Okay, clear. Yes, protoplast will be the right. Sorry, hybrid protoplast is the right answer. Which of the following two organelles contain their own genetic material, DNA and ribosome? Simple question. What are the two organelles? Semi-autonomous organelles. What will be the two organelle? Yes, any guesses? Yes, mitochondria and chloroplast. Yes, excellently. It will have its own genomic DNA. Sorry, own DNA will be there. Apart from that, own ribosome will be there for synthesis of their proteins. Next, the enzyme responsible for initial CO2 fixation in C4 plants. They are asking what will be the initial CO2 fixation enzyme in C4 plants. What will be the answer? Rubisco or PEP carboxylase? Yes, PEP carboxylase is the enzyme involved for CO2 fixation in C4 plants. In C3 plants means Rubisco will come. Okay, Rubisco will come for C3 plants. Next, simple thing only they are asking about the water structure. This we have seen very briefly in our class. They are asking about water structure. Water molecule consists of an oxygen atom covalently bound to two hydrogen atoms with no net charge. They are asking water molecule will have an oxygen atom that is covalently bound to two hydrogen atom with no net charge. No net charge. Second option they are giving because oxygen atom is more electropositive. They are saying this is electropositive. It tends to attract the electron atoms of covalent bonds, resulting in partial negative charge on hydrogen atom. Whether this will be clear? Whether this will be correct? No. This is an even, it's an electronegative. Oxygen in an water molecule is, it's an electronegative atom, and the most important thing, it will takes the H, this will form a covalent bond because of this electron sharing. Yes, yes, because of this electron sharing, this oxygen will have an what? A negative charge will be there. This hydrogen will always have a positive charge. Okay, so statement 2 is, it's a wrong statement because even though it is electropositive, this will attract the electron, but negative charge will be there on the oxygen and the positive charge will be there on hydrogen. So statement 2 is wrong, statement 1 is right. So the right option is option 3. Okay, yes, electronegative, yes, very good. It's a not a electron positive, it's a electron negative. Uday Lakshmi, excellent. Next question. Nitrogen is an example of which following group of nutrient according to biochemical function? Okay, nutrients are classified into 4 major classes. On what basis these nutrients are involved, whether it is involved in enzymatic function or primary function. So group 1 will always comes under primary function, like cellular components, how it is made up of. These things will come under what? Yes, group 1. Nitrogen will always, nitrogen, phosphorus, these things will come under group 1 classification. Excellent. Okay, it's a group 1 classification. Okay, the scientists who created the discovery of cytokinin, plant hormones, scientists who have discovered cytokinin. Cytokinin was discovered by whom? Yes, Skoog. Skoog discovered the cytokinin. Auxin by FW went. Okay, Nelch Bow, ethylene. Clear, ethylene, Nelch Bow. Yes, yes, correct, correct. Next, which of the following alteration of the gene expression is not an example of epigenetic change? See here, they have asked 2 questions from epigenetics last year. Which of the following alteration of gene expression is not an example of epigenetic change? They are asking. What will be the answer? What will be the answer? Yes. Phosphorylation by kinases is not an example, but methylation of the DNA that may influence the gene expression, modification of histones, H3K4 methylation, these all are epigenetic modification. Yes. So this phosphorylation by kinase is not an epigenetic modification. Okay. Which of the following catalyze the change in linking number of DNA molecule? They are saying the linking number will be changed by which enzyme? Linking number is changed by which enzyme? Helicase or topoisomerase, telomerase or ligase? Just use your brain properly. Helicase, we know what it will do. Double stranded DNA will be converted into single stranded. Okay. For strand separation, this helicase will be done. Yes. Topoisomerase, this will be involved in changing the linking number. Telomerase for what? It's for end replication in eukaryotic genomes. Ligases, what it will do? It will used for ligation of the fragments. Okay, so the right option is topoisomerase. Okay. Another most important question. This question they have asked 3 times repeatedly. Okay, 2024, 23, and 25. Okay. Which of the following is not a variant of blast? There are different variants of blast are there. Okay. First we will see that one. There are different variants of blast are there. Okay. Blast N, blast P, blast X, T blast N and T blast X. Okay. These are the different variants. Total 5 variants of blast are there. Okay. Here the query type, what we are like doing. Okay. Input sequence will be whether DNA, protein or whatever it will be there here. Then subject type will be there, whether DNA, protein or DNA. Okay. In BLAST N, the query type will be DNA and the subject type will also be DNA. Clear? So BLAST N, N for nucleotide. So nucleotide will be there. Here also nucleotide will be there. BLAST P, P for protein. Protein will be there. Here also protein will be there. Okay. BLAST X, you see here BLAST X will have nucleotide will be there, but the subject type will be protein. Then see here another thing, T blast N and T blast X. This is another important thing and confusing also. T blast N, T blast X. Okay. T blast N protein sequence will be the query and subject will be the DNA. Okay. So T blast X DNA will be the query type and DNA will be the subject type. This you have to memorize properly. From this they will ask the question. Now you tell me the answer. Which of the following is not a variant of blast? Which of the following is not a variant of blast? We have already seen the 5 variants. Which is not the option? Variants of blast. Yes. Blast N we have already seen. Yes, excellent. Blast N we have seen. Blast P also there. Blast X also there. But T blast NX is not there. So this is the wrong option. Yes. Next, what is the genetic material of Gemini virus? What is the genetic material of Gemini virus? Whether it's a single stranded RNA or double stranded DNA or double stranded RNA, you have to know. You have to just see. You have to just read what are the example of double stranded DNA virus, single stranded DNA virus, and single stranded RNA virus. Okay, these are most important thing. They will ask at least one question from this. Okay, Gemini virus is a typical example of single stranded DNA virus. Okay, it's an single stranded. Yes, it's a single stranded DNA virus. Then what will be the example of double stranded DNA virus? Cauliflower mosaic virus. Okay. Then single stranded RNA example virus is what? TMV, tobacco mosaic virus. Okay. These are the most common questions. Okay. So Gemini virus is an example of single stranded DNA virus. Okay. This is another important thing. T. Out of the amount of water absorbed by plant roots, if plant is absorbing some of the water, What quantity will remains in the plant to support its growth? Okay, what amount will be remains in it to support its growth? For example, 100% of water is absorbing. Yes, excellent. Only less than 1% or less than 1% will be available for supporting plant growth. This is another important thing. Only 1% will be available. Okay. Next, they have given different cell organelles along with the marker enzymes. Okay. Nucleolus. Nucleolus. We know nucleolus primarily ribosomal RNA synthesis will be happen there. So A will come. RNA synthesis will be there. Yes. Ribosomal RNA synthesis will be there. Chloroplast, what it will happen in chloroplast? Any guesses? Chloroplast, what it will happen? Plasma membrane and peroxisome. Plasma membrane and peroxisome. Here options are wrong. You match and let me know. Yes. Peroxisome catalase will be there. This is clear. Yes. Peroxisome catalase will be there. Yes. Next thing. Plasma membrane. Chloroplast is the major site for what? It's a major site for nitrate reductase. This is clear, na? Chloroplast is the major site for nitrite reductase. Nitrite reductase. So, this will come option B. Okay. Chloroplast will come nitrite reductase. Plasma membrane, what it will do? Plasma membrane. It will have a membrane bound receptors. This phytochrome will have alpha 2 structure. This will be there here only. Okay. Nucleolus, ribosomal RNA synthesis will be happened here. This is clear. This is clear. CD. DCBA. Okay. DCBA. This will be the right option. You just see this one. Okay. This is the right option. Okay. Next. The quality of DNA in an extract can be determined by measuring its absorbance at 280 to 280 nanometer and calculating the ratio of 280 to 280 ratio. Okay. This is correct only. The quality of the DNA we will quantify using the ratio of 260 to 280 using nano drop. Okay. Nano drop. This is simple thing. If the ratio is 1.8 means the DNA is pure. If greater than 1.8 means RNA contamination will be there. If less than 1.8 means protein contamination will be there, or else phenol contamination will be there. Okay. So first statement is correct. The 260 to 280 ratio, if it is less than 1.6, indicates the presence of proteins, phenols and other contaminants in the DNA extract. What will be the correct answer? What will be the correct answer? Yeah, both are correct statement only. If the value is 1.6 means that will be the indication of phenol and protein contamination. So both the statements are correct. Both statement 1 and 2 are correct. Okay. Which of the following is a protein-protein interaction method? Any guesses? Which is the example of protein-protein interaction method? RT-PCR or qRT-PCR, split ubiquitin or Y2HSA or Y1HSA? This we have already seen, yes. Y2H, yes, excellent. Y2H is Y2H we will commonly use for protein-protein interaction study. This we already seen, protein-protein interaction study we will done. Y1H for protein DNA interaction and Y3H for protein RNA interaction. This we have already seen clearly in our lecture series. Yes. Next, they have given same thing only. Same thing they have given. Okay, again, same question. This we have already discussed. Now, the end product of light dependent reaction of photosynthesis is what? End product of light dependent reaction of photosynthesis. Yes, oxygen will be there. NADPH2 will be there. Apart from that, ATP will be synthesized as a end product of light dependent reactions in photosynthesis. Next, they have given different crops. Yeah, different crops they have given, and state with maximum production. General agri question. State with maximum production. Different crop and state with maximum production. Castor production will be always more in where? Gujarat for G2. Okay, G2. Okay. Sunflower production will be more in Karnataka. Okay. Yes, Kenkam. Sunflower production will be more in Karnataka. Mustard, where? Rajasthan will be there for mustard. Okay. Soybean, this simple thing. Soybean, we everyone knows soybean production will be more in Madhya Pradesh. So soybean is Madhya Pradesh. Option D will come 3. So this is the right option. Okay. If you know any one of the things, easily we can eliminate and we can select the option. Yes, yes. Udaya, yes, correct. Soybean is Madhya Pradesh. Okay. Example of unsaturated fatty acid. What is the example of unsaturated fatty acid? Stearic acid, oleic acid, lauric acid, palmitic acid. What is the example of unsaturated fatty acid? Yes, stearic acid, 18 carbon will be there with 0 double bonds. Oleic acid, 18, 1 double bond will be there at del 9. Lauric acid, 12 carbon will be there with 0 double bond. Palmitic acid, 16 carbon will be there with 0 double bond. So, what will be the answer? Only oleic acid. Only oleic acid is the option B is the correct example of unsaturated fatty acid. Clear? Yes, excellent. Udaya, excellent. Option A is the right option. Remaining 3 are what? They are saturated fatty acid. Okay, lauric acid, oleic acid, lauric acid, myristic acid, palmitic acid, and stearic acid. These 4 are major example. Okay, LYMPS. You just remember LYMPS. Okay, lauric acid, myristic acid, palmitic acid, and stearic acid. Number of carbon 12, 14, 16 and 18. Okay, these all are example of saturated fatty acids. Okay, so only B is the right option. Yes. See here they have given different chemicals and inhibitors and they have given what will be the major role. Okay. We know seed viability. We will always use tetrazolium test for checking the viability of the seeds. This is a very simple question. So A will come option 4. Wherever it is coming, A will come option 4. Yes. Chloroplast assay or Hill reaction. The inhibitor for Hill reaction is what? DNP they will use. Dinitrophenol. This will act as an inhibitor. Anti-transparent. Hexadecanol will be used as an anti-transparent. Respiration inhibitor is a rotinone. So this is direct question. This will be the right option. Next, about markers they asked. Okay, markers they asked. Okay. You see the option itself, SSRR multi-allelic marker, SSRR codominant marker, dominant. See, these both are contrasting. Codominant and dominant will not come in a single marker. So here some mistake is there. Then SSR acronym is sample sequence repeats. Wrong. It's a simple sequence repeat. So option D is wrong. Wherever option D is coming, that is wrong. This is clear first thing. Second thing, SSR is always it's an codominant marker. So option C is also wrong. So wherever C is coming, that is wrong. So the right option is what? Option A and B. SSR is an multi-allelic marker and SSR is a codominant marker. Clear, students? Clear? Last question of today's section. Okay, just observe the question properly. In the era of global boiling and beyond, it is expected there will be an acidification of the world's ocean. Okay, general agri question. Okay, acidification of world ocean. Okay, in the era of global boiling and beyond, it is expected that the growth of shell building the organism will positively impacted due to the abundant availability of carbonate precursors in the ocean. See, they are saying, we know at present the carbon dioxide concentration in the atmosphere is increasing. It's around 0.4. Okay. around 440 ppm is there. Okay, 440 ppm is there now. Okay. So if the concentration of CO2 is increased, this can act with water in the ocean that may lead to formation of carbonic acid. This is clear that ocean can absorb the carbon dioxide that will lead to the formation of carbonic acid. So this is true. Expected that there will be a acidification of the world's ocean because of the CO2 absorption. Carbonic acid will form that can be lead to acidification. So statement 1 is correct. Then second statement what they are saying means because of this process there will be a huge amount of carbon available for these coral reefs and shells for their growth. This is wrong. Why means the available carbonates will become decreased that may severely affects the growth of these coral shells. Okay. So statement 2 is it's a wrong statement. So the right option is option 1 is correct and option 2 is incorrect. Statement 1 is correct and statement 2 is incorrect. Any doubt students? With this we are completing our lecture. Okay. Next class we will see at 2:30. Okay students. Thank you. We will see you at 2:30. Remaining things. Huh? Live stream end.